times. All right, welcome everyone. So today I'm going to talk a bit about the work that my colleagues and I have been doing on the role of gut microbes in ant evolution. I'm fortunate to have several of my colleagues here. I just saw Scott Powell walk into the room. I know Jake Russell's at least at the conference, if not in the room. And unfortunately, John Wirtz couldn't be here, but he's a key component of this work. And so why is it that we'd actually want to study the gut microbiota of ants? I would actually argue ants are an excellent system to address questions about the evolution of gut-associated microbes, because there's a lot of interesting aspects of their biology that makes them interesting. They're incredibly species-rich, they have a broad distribution, and we do know that there's some influence of gut-associated bacteria across the group. And so specifically, one of the things that we've been trying to address is what's the potential role of these gut bacteria in diet and nutrition. And so this is the sort of fundamental question that I'll be talking about today, but there are some interrelated aspects of this that I won't necessarily be able to touch on, but that we hope to be able to accomplish during the aspects of this project that are funded by the National Science Foundation. And one of which is asking the questions of transmission. And so it's related to this idea, if bacteria are important to diet and nutrition, how are they transmitted among individuals? And not just individuals that are interacting within a colony, but those same individuals that are interacting in an environment, and then how does that play out through evolutionary time? Next, if they are sort of playing out through that evolutionary scale, when would we expect to see lineages that are co-evolving, and when will we not? So you can imagine that there are some bacteria that may be just transient, and we wouldn't necessarily expect to see coevolution. There are some bacteria that may be, in fact, pathogenic in the gut, and so we may or may not expect to see that. And for those that are potentially upregulating hosts' diet, maybe we really would expect to see things like co-diversification or co-speciation. And lastly, one of the things that's really nice is that because ants have such a broad distribution, we can ask the question, what role does geography play in this? When would we expect to see microbes just acquired from the environment in which the hosts are found? And when would we expect to see maybe a convergence on those bacterial communities through time? Now I'm going to spend most of the talk talking about the work we've been doing on diet and nutrition. And this begs the question, if they are in fact upregulating their host's nutrition, what's the mechanism behind that? And so there's sort of three broad categories that people have thought about, and one of which is maybe it's through something like nitrogen fixation. So this is what we see in a lot of kinds of plant systems. It's been observed in other kinds of insect bacterial associated systems as well. Of course, there's potential for things like nitrogen recycling as well as nitrogen upgrading. So starting to be able to tease those apart is a potential mechanism within the host. Now, as I already alluded to, I have a great group of collaborators, and I'm not going to be able to spend a lot of time on the work of John Wirtz, but what's really nice is he's been able to come in and bring his culturing expertise and be able to do metabolic assays to say, what are the functional capabilities of those microbes that we're finding within the guts? And so what we do is we go out in the field, bring ants back, and he isolates the bacteria from their guts, and we can actually start to ask questions what they're in fact doing. And he's got some really interesting results. I'm not going to be able to present them today. They're sort of in the early stages, but keep your eyes out. Now, the other aspect of this work actually centers around a lot of the work that Scott Powell's been doing for quite a long time in this system. And that's not just looking at the species diversity, but actually asking questions about the functional ecology of these ants, how they're interacting with one another, and how that might be a mechanism for exchange of bacterial communities across both species that are co-inhabiting the same tree, but also through time. Now, this work is also sort of riding on the work of two postdocs that are associated with the project, one of which is Shauna Price, who's done some really elegant work, and she's actually speaking directly after me. And she's been looking at the evolution of the host ants themselves, and the work she'll be presenting today is sort of on the evolution of morphology related to them. But on the flip side of that is we have John Sander, who's been looking at the gut-associated bacteria within the group and asking questions about co-diversification. So this sort of gives us a nice framework to begin to address these questions. So when my colleague Jake Russell and I first started working on gut-associated microbes across ants, it was quite some time ago. And really what we were interested in was understanding something about the diversity associated with ant hosts. And so at that time, really what we had to do was a lot of cloning and sequencing. It was a lot of arduous work. I will give Jake Russell all the credit. I see him in the back of the room. And really what we did is we just asked what bacteria do we find associated with ants. And so in this study, we sampled almost 400 ants from 150 ant genera. And maybe not surprising, what we saw is a lot of diversity. But what jumped out to us was this lineage of bacteria from the rhizobiales. Now, some of you might be familiar with other bacteria from this lineage, because these are the bacteria that are associated with root nodules of leguminous plants, where we know that they're fixing atmospheric nitrogen for their plant hosts. So because we're finding a related group of bacteria in the ants, is it a similar mechanism? Is that maybe how they're upregulating their host's diet? And I'll come back to that near the end of the talk. we ask the question, when we find this group of bacteria associated with hosts, and we use stable isotopes, in this case it's the ratio of heavy to light nitrogen, to infer where they're feeding on the trophic scale, we can see that there's a high association or infection with rhizobiales for ants that are feeding low on the trophic scale. So each of those dots is an ant genus, represented by one or more species and individuals, and we've inferred where they sit on that trophic scale. Now you notice that only individuals that are feeding low on the trophic scale have an association with rhizobiales, but you may have also noticed that there's two dots that are sitting at the low, the zero frequency with rhizobiales, but are feeding very low on the trophic scale. And it turns out that those are the carpenter ants and their allies. And we already know that this group of ants have specialized bacteria and bacteriocytes that are upregulating their host's diet. So essentially what we're seeing is that we're seeing nutrient provisioning potentially through either Blockmania or rhizobiales, suggesting that there's potentially some association of these bacteria with the host. Now we wanted to understand something about what are the mechanisms and sort of where are these groups of bacteria distributed. So I'm going to present several lines of indirect evidence before walking into some of the more direct evidence. We wanted to understand what sort of is acquired from the environment. So to do that, we survey all the bacteria associated with the legs of the ants. To control for bacteria that may be introduced into the digestive tract but maybe don't become resident members, we sample the mouth cavity. We then sample 3 distinct parts of the digestive tract, the crop, the midgut, and the hindgut, and ask what bacteria do we find there. And just sort of as a reminder, the crop is essentially a social stomach, so it's where food is stored but never processed. And so it's similar to what we see in birds where they have the social trophallaxis or food exchange. Now, once you move into the midgut and the hindgut, that's where we begin to see digestion occur. So here what we're using is next generation sequencing to survey the entire bacterial community, so not a specific lineage. And what I'm presenting here is a PCOA of that entire bacterial community. And what you'll notice is all those samples that came from the leg, the mouth, or the social stomach seem to have no similarity among all of those individuals that were sampled. It's only once we move into the hindgut and the midgut, those bacterial communities become highly similar, suggesting that there's something driving the community structure in these regions. So it's another sort of indirect line of evidence that there might be some functional role of these microbes associated with diet or nutrition. Another really nice piece of work actually came out from Jake Russell's lab by a postdoc, Peter Lukasik, where he used in situ hybridization to essentially demonstrate where do we see the highest density of bacteria within the gut. And interestingly, what we see is that the bacteria are associated with the part of the digestive system where the Malpighian tubules, which essentially deposit all of the uric waste into the digestive system. So if urea is the building block for sort of upregulating the host's diet, we essentially have the mechanism for it being delivered specifically to the region with the highest density of bacterial communities. Now, Yi, who was a graduate student in Jake's lab, who's now a postdoc, actually did some really nice work where she essentially said, well, what happens when we feed the ants on diets that mirror what we know that they eat in nature? So the group of ants that we work on, the turtle ants, have been observed to eat 3 things. Plant-derived resources, usually things through extrafloronectaries or by tending sap-producing insects. Again, very low in nitrogenous resources. We also know that they will go to bird droppings, which unfortunately is an ephemeral source of food and one that ants can't rely on, but is very rich in nitrogen. So if you're nitrogen deprived, you actually may be highly attracted to this food resource. We also know that these ants actually feed on pollen, and they're one of the few species that take advantage of this food-rich resource. And part of the reason that so few things actually eat pollen is that it's heavily defended by its pectin cell wall, which is why it fossilizes so well. So one of the only other groups that actually takes advantage of eating pollen are the bees. So Nancy Moran's group has done some really nice work demonstrating that in honeybees, the gut bacteria can actually lyse through that pectin cell wall, then releasing the nutrients. Now, when she did these diet experiments and looked at the bacterial communities, what we see is we see a significant increase in the bacterial communities, or shifts, in the proportion of some lineages when they're fed on pollen-based diets, suggesting that there may be some other mechanism there. And interestingly, one of those plays is the rhizobiales. Now, following up on that work, she did some really nice work where she did some tests of nutrient provisioning. So in this case, what she did, she took colonies and split them, and she fed half with antibiotics, so essentially curing them of their gut-associated bacteria, and then left some intact. She fed them on diets that had labeled amino acids, so we could actually ask the question, labeled urea, I apologize, and ask what happens. And interestingly, we only see an upregulation of all the essential amino acids when we leave their bacterial communities intact and have the labeled urea present, suggesting that there is some mechanism there for potentially nitrogen recycling by these bacteria that are left in the gut intact. Now, another line of direct evidence actually comes from the metagenomic work that they've been doing. So what we've been doing here is to sort of sampling all of the bacteria associated with their guts and then reassembling the communities. And what we see is that there are essentially amino acid biosynthetic pathways that are complementary between the ant's own genome and the bacterial genomes that we find associated with this. And in this case, then we find evidence for the synthesis of all 20 essential amino acids. Again, saying that there's something about having this bacterial community that's stable and persistent through time, which we know because we've done diet experiments where we fed them in the laboratory for up to a year to show that it's not something they're just acquiring from the environment. So thinking about what are the broader implications for this. So in the case of ants, it does appear that we're seeing some bacterial provisioning into the ant host themselves. Now, I started the talk with talking about the group of bacteria, the rhizobiales, and saying that, well, in this group of bacteria, we know that in some cases they upregulate their host plant's nutrition through things like nitrogen fixation. We've actually done acetylene reduction assays in 2 different species of turtle ants, and we find no evidence for that happening within the hosts. So we don't believe that this is the mechanism that's happening, although it has been demonstrated in other groups of ants. Now, through the feeding experiments with the labeled urea, as well as with the sequencing of the metagenomics, we do in fact see evidence for nitrogen recycling, and we suspect that this may be the mechanism for upregulating host nutrition within the turtle ants. But we can't rule out that there still might be some amount of nitrogen upgrading happening. But so this is one of those interesting systems where we're planning to pursue this further. So now we have this, putative, association. Now we have the ability to culture these microbes, and we can ask what are the capabilities, the functional capabilities and metabolic capabilities of those bacteria themselves. And ultimately what we'd like to be able to do is then do feeding experiments where we reintroduce them into the host once we've cured them of their host bacterial associations. So ultimately this work really couldn't be done if it wasn't for the Dimensions of Biodiversity grant from the National Science Foundation and my awesome colleagues and collaborators, both on this work, but mostly through the Dimensions program itself and the members of my lab. And I think there's time for a few questions.